The study of protein folding is one of the most complex and fascinating areas of research. Despite the fact that we've been at it for well over 60 years, it still feels like we don't know that much about protein folding. So many scientists have spent a lifetime studying it, and yet we still dub protein folding as the protein folding problem. I think this graphic abstract says it really well. We literally actually don't fully understand how an unfolded protein on the left, a simple polypeptide chain, becomes a fully folded, native and functional protein. We do know some things. We do know that protein folding is rapid, that it takes less than a second, and that it doesn't involve a random conformational search. How do we know that? Well, if you took a very small protein, 100 amino acids in length, if it went through a random search, that is, it tried all of the different possible conformations, it would take 10 to the 87th seconds. To give you perspective, that is more seconds than the universe is old. So randomness is out of the question. And in fact, we probably would have hypothesized that based on our current existing knowledge. Remember talking about steric preferences for certain amino acids to exist in particular spaces, and the fact that phi and psi angles are constrained. So even with our knowledge, we would have said, yeah, I bet it's not a completely random search. We also know that protein folding is cooperative. That is to say that amino acids in particular local neighborhoods act together to form interactions, but also holistically within three-dimensional space, we know that there's a cooperativity about the way in which interactions form. And we know that there is a protagonist in protein folding. That protagonist, as you might have guessed, is water. And in fact, this idea that proteins have sort of an essence, sort of a nucleating kernel that is maybe even present at the moment of synthesis, and that allows for sort of this immediate collapse, and that is in response to water. Well, that's been an idea that's been out there and strongly held, this idea of hydrophobic collapse and this domino effect where interacting neighborhoods, it's cooperative and it's cooperative in 3D space, and that gives rise to the formation of this native state. However, there's a lot of debate, and there's a lot that we don't know about those proposed ways by which an unfolded protein becomes a folded protein. And in fact, quite honestly, there's a debate about unfolded and what that means. We've talked about with our conversation of ribonuclease A, pictured behind me, and Christian Anfinson's work, this idea of denatured. And in fact, there's a difference between unfolded and denatured. And denatured, we generally define that as losing function. Now, there may be local areas that retain some sort of structure and even resemble structure of a native form, but it doesn't function. The protein doesn't retain its function. Whereas maybe unfolded could be defined as what we would see when a protein is present in the presence of a high concentration of denaturants, and it's completely just its linear form, completely unraveled, no structural elements retained. But truthfully, that's not likely to be what happens immediately after synthesis. And so this idea of studying, in fact, because really that's not an in vivo argument that we would see it in this presence of high concentration of denaturants and just in its linear structure form with just its primary structure. So scientists have long debated whether the transition from unfolded to denatured might be valuable in understanding what forms this nucleating core, and whether it's just a hydrophobic collapse or whether there is more to that. One other interesting thing that researchers have studied to help them understand that is to look at mutating particular amino acids within the primary sequence, and then looking at how that impacts on folding. And that's really helped them to understand better and to give name to the nucleation condensation mechanism. So this idea of folding is driven by nucleation of certain residues, but formation of the nucleating core does require that there's a significant amount of the remaining protein that is in a conformation resembling or on its way to native. So we could say formation of the nucleus, nucleation is coupled to a more general formation of structure, condensation, giving rise to sort of this diffuse transition state that resembles the native state. When a protein folds, it experiences these sort of constant counter interactions between configurational entropy and effective energy. Effective energy is driving folding to the low energy native state, but of course, configurational entropy is fighting against that. So when a protein folds, it is going through the process of falling down, we sometimes say, this energy well. So as it does so, the number of accessible configurations, which determines the configurational entropy, decreases as the native state of the protein is approached. So we see that at the quote bottom of the energy well. So it's like there's not one path to folding to this native state, but instead there is an increasing probability, and there's a reasonable number of paths and conformations that can lead to the folded state, despite there being sort of these local energy minima that are improperly folded or partially folded state that eventually falling to that favorable native state occurs. I like to think about this by an analogy. So I actually hate puzzles. And one time we were on vacation, and we were in this vacation rental up in Montana. My mom was doing a puzzle, and she's like, "Rachel, you need to join us in doing this puzzle." And I was like, "No, I really am enjoying studying protein folding." So instead I had my nose in my biochemistry textbook, and I was studying protein folding. And so it was a good deal. She was doing what she loved, I was doing what I love. And then a little while later, she yells out, Rachel, you've got to come help us. The more pieces I put in, the easier it gets to put in pieces. And it was like, in that moment, I was like, eureka, that's protein folding. Because the more cooperative interactions that take place, the more likely the protein is to find itself in its native state. So this cooperativity that she was seeing in doing the puzzle, right? The more pieces she put in, the easier it got to put in pieces. So it was, and the key thing in that is also true of protein folding is that that correct interactions can't be undone. So once you put in a piece that fits, you can't take that out, and it makes it easier to put in pieces. And this is the same thing that we see in protein folding. So I invite you to read both of the articles that I cited in this little mini lecture. They go into far more depth, as does the Jakubowski text, if you're interested, and that's open access and ready for you to read. So cheers to protein folding.